Welcome to SS2 Biology. Today we explore one of the most important chemical processes on Earth, photosynthesis. Every plant you see around you, from the mango tree in your compound to the cassava in the farm, uses sunlight to manufacture food. Without photosynthesis, there would be no oxygen in our atmosphere, no food chains, and no life as we know it. Photosynthesis is the process by which green plants, algae, and some bacteria convert light energy into chemical energy stored in glucose. The overall equation is carbon dioxide plus water, in the presence of light and chlorophyll, produces glucose and oxygen. In this lesson, we will divide photosynthesis into its two major stages, the light-dependent reactions and the light-independent reactions, also called the dark reactions or the Calvin cycle. Understanding these two stages will help you answer WAEC questions accurately and give you a deeper appreciation of how the natural world sustains itself. The light-dependent reactions occur in the thylakoid membranes of the chloroplast. When sunlight strikes chlorophyll molecules, the light energy excites electrons to a higher energy level. These energized electrons travel along the electron transport chain, releasing energy that is used to produce ATP, adenosine triphosphate, and NADPH. At the same time, water molecules are split in a process called photolysis. The splitting of water releases oxygen as a byproduct. This is the oxygen we breathe. The equation for the light reactions can be summarized as light energy plus water produces ATP, NADPH, and oxygen. Two photosystems are involved. Photosystem 2 absorbs light at 680 nanometers and is responsible for splitting water. Photosystem I absorbs light at 700 nanometers and is responsible for reducing NADP+ to NADPH. The ATP and NADPH produced here will power the next stage, the dark reactions. Despite the name, dark reactions do not require darkness, they simply do not directly need light. They occur in the stroma of the chloroplast. The Calvin cycle uses the ATP and NADPH from the light reactions to fix carbon dioxide into organic molecules. The cycle has three stages. First, carbon fixation. Carbon dioxide from the air combines with a 5-carbon compound called RuBP, ribulose bisphosphate, using the enzyme RuBCO, producing two 3-carbon molecules called GP, or glycerate 3-phosphate. Second, reduction. ATP and NADPH are used to convert GP into GLP, a glyceraldehyde 3-phosphate, a 3-carbon sugar. Third, regeneration. Most GLP molecules are used to regenerate RuBP so the cycle can continue, while a small fraction is used to synthesize glucose and other organic compounds. For every 3 turns of the Calvin cycle, one 3-carbon sugar is exported for use in building glucose. In your examinations, you may be asked to name the products of the light reactions, the location of the Calvin cycle, or the role of RuBCO. Know this well. Several factors affect the rate of photosynthesis. Light intensity. As light intensity increases, the rate of photosynthesis increases up to a saturation point. Carbon dioxide concentration. More CO2 means faster Calvin cycle activity. Temperature. Enzymes like RuBCO work best at optimum temperatures, too hot or too cold, and the rate drops. Chlorophyll concentration. Plants deficient in magnesium, needed to make chlorophyll, photosynthesize poorly. In Nigeria, farmers understand this practically. Crops grown in well-lit, fertilized farms with adequate water produce far more than shaded, nutrient-poor plants. WAEC tip. Be able to draw and label a graph showing how light intensity affects photosynthesis, identify the limiting factor at each point, and write the balanced equation, 6CO2 plus 6H2OC 6H12O6 plus 6O2. Know the roles of ATP and NADPH and be able to distinguish the location of light reactions from dark reactions.